Cool. Thank you guys very much for having me. Thanks for the kind introduction. So today I'm going to be talking to you about the research that I've been doing over the last number of years, which encompasses genes, brains, and neurodegeneration. So firstly, just a little bit about my background as how I got to where I am today. So I started off my studies at the University of Manchester, where I studied physics, and I really enjoyed kind of understanding the fundamental principles that govern our universe. However, I was always found that I was drawn towards neuroscience, and so in my free time, I was taking a lot of classes related to neuroscience, as well as kind of watching online lectures. But unfortunately, in the UK, our curriculum is a little bit restricted, and so you're really not able to take formal courseware in other disciplines. But luckily, I was able to study abroad for 1 year at UCLA, where I was able to have a bit more broader scope. So I did mainly physics, but I was able to take a couple of cognitive neuroscience classes, where one of the professors encouraged me that if I wanted to develop more of a focus towards neuroscience, that I ought to go through neuroimaging, given my background in physics. And so I took his advice, and when I came back to Manchester, I completed a master's project, which looked at biophysical modeling of the fusion MRI signal. I really enjoyed that work. It really kind of grabbed my attention in terms of really wanting to engage in neuroscience research. So I took this experience and I worked as a research assistant for a couple of years at University College London, where I worked on prediction models predicting language outcome of stroke patients. And so I'm going to talk to you a little bit about this work and midway through the talk. And then ultimately, I took this experience and came to UCSD to study cognitive science as a PhD, where I not only combined neuroimaging, but also developed expertise in human genetics. So for human genetics, why might we be interested in studying human genetics in the context of the brain? Well, twin studies have, over the last 50 years, established that genetics contributes pervasively to traits related to brain and behavior. So heritability is kind of a critical metric that often gets computed, which relates to the amount that we can attribute genetics to various traits. And so here are a number of traits related to the brain, and you can see the heritability kind of goes from about 40 to about 90% for each of these different brain traits. So if we can understand the genetics giving rise to traits, then we may understand and uncover the biological mechanisms giving rise to those traits. And when the trait being studied is a disease, investigating its genetics may lead to therapies or to cures. A particular pertinent example, I think, is spinal muscular atrophy, which is a very damaging genetic disorder, in which individuals who have this in its most severe form don't survive past 1 or 2 years of age due to the pathogenic nature of how it damages motor neurons. But by understanding the genetics of this disorder, drugs and therapies have been developed. So Zolgensma is one from a few years ago, which for these individuals, it's effectively the closest thing that we could describe to as a cure. So individuals that would have died, let's say 1 or 2 years of age, now live well beyond that time point, and are able to reach major milestones that they never would have been able to without development of this drug. And so here I'm really making a case for studying genetics, but it's really important, as I'm sure many of you are aware, kind of the reciprocal of this nature is that, to some degree there's genetics, but what isn't explained by genetics is explained by environment. And so there really is a substantial contribution coming from environment that's influencing many of the traits that we study and care about that relate to brain and disorders of the brain. And so for this talk, I'm going to be talking to you about a number of projects looking at genetic influence of behaviors and disorders of the brain. Then I'll be talking to you about a project which looks at trying to use genetics to develop neuroimaging biomarkers that can use kind of inform risk for disorders of the brain, particularly in the context of studying Parkinson's disease. And then finally, I just want to reiterate that environment is a really important component here, and so it kind of shouldn't be neglected in the context of the results of the project. And so for a lot of my work, I've kind of worked at sort of two ends of the lifespan. So for a lot of my PhD, I did work looking at the genetic influence of adolescents. So I'm going to briefly talk to you quite quickly about that work. But then I want to move on to talk to you more in depth related to projects which kind of form the main focus of my research agenda currently, which is looking at later in life, looking at neurodegenerative disease and neurological conditions. So firstly, looking at the genetic influence in adolescence, I'm going to talk to you about a few projects which analyze data from the Adolescent Brain Cognitive Development Study, which is a longitudinal study of just short of 12,000 children aged 9 to 10 years old. And this includes multiple data types, including genetics, neurocognitive assessments, mental health assessments, as well as substance use questionnaires and demographics. Something I want to highlight here is that I serve as the principal bioinformatician responsible for curating, processing the genetics across the entire sample. And so I'm going to talk to you briefly about 3 projects here. Firstly, looking at the genetic influence of cognitive functioning in this sample. Next, looking at the genetic influence related to psychiatric risk, and finally looking at substance abuse risk in this sample. So firstly, focusing on cognitive functioning, neurocognition is often studied. There are 2 kind of major factors that people are often interested in, one being crystallized performance. So this is more related to cognition in that's related to acquired knowledge. So a typical task might be one's vocabulary or reading comprehension. And this is often contrasted with fluid comprehension, which is thought to be more related to cognition in novel situations. So here is a rated progressive matrix where individuals look at the first row, the second row, and given that sequence, they're asked to complete the third row. And traditional theories of intelligence would predict that the genetic influence on crystallized measures should be less than that of fluid measures, because crystallized cognition is more related to one's environment. So it'd be thought that an environment should have more of an influence in terms of the variability that gets created for crystallized cognition. However, a recent study in adults found the reverse. So here on the y-axis are tasks in increasing order of their kind of cultural dependence. So kind of these more crystallized measures tend to be more heritable. So they tend to have a larger genetic contribution. They found this in adults as a kind of counterintuitive finding, but they weren't able to establish it in children. And so we sought to look at this in this ABCD study to see if this was something that we could find. The way in which we did this was we generated what's known as a polygenic score of intelligence, where we leveraged the previous study looking at a genome-wide association study of 270,000 individuals for whom genetics were measured, and performance on intelligence measures was measured, and then models were fit to associate genetic variants to performance on these tests. We then took the ABCD individuals for whom we have genetics, and then we took these weights and used them to calculate kind of variability on how we predicted they may have performed on this same task. And so for this study, we looked to associate this score with variability on crystallized and fluid measures of intelligence, with our prediction being that the polygenic score would be more associated with crystallized performance. And what we found was that was the case. So here on the x-axis are different measures that are crystallized in red and fluid in blue. And on the y-axis is how strongly that polygenic score is associated with performance. And we have each of the subscales that make up performance on those tests here in red and blue. And so we found that the polygenic score was correlated with all neurocognitive measures, but critically that the polygenic score was more associated with crystallized rather than fluid measures. And this is in contrary to what traditional theories might have predicted, but in agreement with this recent study in adults. And so one explanation which we think might be kind of driving this relationship is that it may be explained by gene experience correlation, in which individuals who have kind of initial propensities towards performing slightly better on crystallized tasks end up in these environments that further kind of cultivate and amplify those abilities in a way that leads us to kind of overestimate the degree to which genetics is contributing to those traits in a way that it's not kind of so facilitated in fluid measures. And so in that sense, we sort of see this as sort of a case against genetic determinism, in that we believe that there is kind of this critical interplay, which we're possibly not modeling, of genetics interacting with one's environment. And so for this next project, we also looked at the genetics for psychiatric risk, which is psychiatric conditions form a huge burden on those individuals, families, and society as a whole. And so if we can identify early life risk factors for intervention, this could be a huge benefit of changing individual outcomes. And so in this young sample of 9 to 10 years old and 10 years of age children, what we did was generated polygenic scores predicting risk for a number of psychiatric conditions. and then evaluated those across mental health assessments that we had in the sample. So here are each of the polygenic scores on the left-hand side, and then on the top is the huge battery of mental health assessments that we have in the sample. Some are given to caregiver and some are given to youth, but both are assessing the youth's mental health state. And so each element showed the variance explained as a percentage, and dots indicate FDR significant association. So there's a number of associations here, but I'm just going to kind of highlight some of the biggest ones. Is that ADHD seems to be related to both internalizing and externalizing symptoms. Genetic risk for depression seems to be associated with more internalizing symptoms, and suicide attempts seem to be related to internalizing and rule breaking subscales of certain assessments. And so here we're kind of seeing that there do seem to be some detectable patterns of genetic risk for some fairly major psychiatric conditions and life outcomes that appear to be detectable at this early age as well. So finally, for this sample, I'm going to talk to you briefly about an ongoing project which is looking at genetic risk for substance abuse in this sample. So here, instead of using polygenic scores to create genetic risk factors related to psychopathology, we're doing genetic risk factors to calculate risk for using certain substances. And so each of those polygenic scores for different substances are here on the x-axis, and they've been grouped by ones that relate to alcohol, to cannabis use, to polygenic risk for coffee, to general risk factors, as well as smoking. And then on the left-hand side, we have self-reported use for a number of substances. And again, this is for individuals who are 9 to 10 years old. And so there's a number of associations which I think are interesting, but something that I wanted to highlight is that we see that regular caffeine consumption seems to be something that is related to higher risk for both alcohol as well as smoking-related polygenic scores. Which I think is pretty interesting given that the sample is so young and relatively drug-naive. And so in summary, for this portion of the talk, I showed you how an intelligence polygenic score is more related to crystallized rather than fluid measures. We think this might be explained by gene experience correlation in a case against genetic determinism. We also showed that genetic risk for ADHD, depression, and suicide attempts was associated with a number of relevant mental health assessments, and the genetic risk for substances was associated with caffeine consumption at 9 to 10 years old. And so these first two results are published in which I was first author over the past couple of years, and then this third paper is currently being written, which I'm senior author.
So the second one, you took a separate data set that showed and did your statistics on to create a polygenic hazard or polygenic risk for ADHD, depression, suicide separately, each one? Or how?
Yeah. So it's separate previous genome-wide association studies with each of those traits. So it's kind of variable. Some of them are in like 20,000 people. I think the suicide attempt was in about 30,000 individuals. Depression was one of the largest that had about 800,000 individuals. So these were all previous studies, and we used those kind of weights and applied them all within the ABCD sample. And so all of these projects relate to the ABCD sample, but they all leverage previous genome-wide association studies.
And you took that raw data that they put out with all their weights and you assembled them with your approach.
Yeah, exactly.
And then put it in.
Yeah. Okay. And so, that kind of concludes the sort of first part that I wanted to talk about, which is still ongoing work, but it's not kind of my major research focus currently. Now I'm just trying to focus more on sort of later life and neurological research projects. I'm going to step away from genetics for a moment and just talk about the work that I did at UCL, which was looking at how we could use neuroimaging to create predictive models for predicting language outcome after. So this was working on a data set known as PLORAS, which stands for Predicting Language Outcome Recovery After Stroke, which is really trying to work on a pressing need that patients have to know their ability to recover their language faculties following a stroke. And so this data set contained just short of 1,000 stroke patients when I was working on it, who were each given a battery of 27 language assessments known as the comprehensive aphasia test, and they have brain MRI scans as well as demographic data. And so one of the measures which we used quite a lot in this data set was known as a spoken picture description task, in which individuals get given this scene, and they're asked to describe the contents over the course of a minute and assess using objective criteria. And if you train models using brain MRI data, as well as time post-stroke, you can do fairly well at predicting people's performance on this task. So here on the x-axis is each individual's actual speech production score, and on the y-axis is their predicted speech production score using brain imaging as well as time post-stroke. And so the brain imaging calls MRI scans. And so you see the variance explained is 0.6, which is fairly good model performance, and we're doing a fairly good job of separating individuals who will and won't recover their language faculties. So this was previous work that was done in the lab, and the work that I was looking to do is to extend this to see, could we take models that were trained within this data set, this academic data set on English patients who are assessed in English, who have MRI scans, and who are largely chronic, to a validation data set of individuals who are assessed and speak a different language, which is Spanish, who are largely acute, and who were scanned using CT scans. And so this work was made possible by tireless work from Diego and Andrea, who were PhD students in the lab who collected this validation cohort. So I'm extremely grateful for the work that they did for this. And so if we do cross-validation using a slightly different model, which I'm not going to go into the detail of, but here doing cross-validation within the British cohort, we can again see that actual and predicted speech production score, we do fairly well in terms of model performance with the variance explained at 0.5, so slightly lower than before, it's a slightly different model. And then if we take the model that was trained within this British cohort and then extend it to the validation set of Chilean patients, we can evaluate how well we do on model performance. So the variance explained is 0.46, which is relatively good, but you'll notice that we kind of have this sort of overestimation, which most individuals seem to kind of higher than this y equals x line. And so we kind of scratched our heads a little bit to think what could be driving this. And one of the things that we thought was important, and so if we formally test this, we see that the prediction errors are significantly worse in this group versus the British group. But getting back to kind of this, what might be driving it, we sort of found that we were overly optimistic in terms of how well we thought they were going to do on these tasks. So one thing which we thought would be important
is higher better.
Was that
higher score is better?
Yeah, higher score is better. Yeah. So this is basically saying we're thinking from imaging that they're going to do better than they actually do. The one thing that we thought was important is something which we'd seen and others documented, which is that stroke lesions don't tend to stay static over time. So we had longitudinal data which we saw systematically that stroke lesions tend to grow over time. And the growth seems to be greatest after the stroke and slows over time, and possible speculated mechanisms of Loughnan degeneration and chronic hyperperfusion. So why might this be important for this current project? Well, if we kind of take a British subject who may have had a stroke at a certain time point, and then over time, we expect that lesion to grow, then at a later time point, we have a scan and a behavioral assessment, which are both kind of in register with one another. And if we contrast this with a Chilean individual, then we have a scan at a certain time point, and then we have a behavioral assessment at a later time point. And during the intervening time, we expect that this lesion is going to grow. And so our scan, which is acutely scanned, is a poor representation of the extent of the lesion at the chronic stage. And so what we did was we trained models within the British cohort for whom we had longitudinal scans to get a sense of what lesion growth looked like, and then applied those to the Chilean cohort to essentially computationally grow out the lesions such that the behavioral assessment and our kind of imputed lesion growth size was in register with one another. And so if we do that, we find that after lesion growth, we find that there is no longer a significant difference in prediction errors between the British cohort and the Chilean cohort, which is encouraging, and it actually relates empirically to the correct amount of lesion growth. So it's not just any amount of lesion growth, but it's specifically the correct amount that corresponds to empirical lesion growth. And additionally, we also kind of wanted to see for individuals in the British cohort, because we had a subset of them where there was a large time between scan and behavioral assessment, just for pragmatic reasons. And so we wanted to see for those individuals, if we computationally grew out their lesions to the correct time point, does that improve our prediction error? We find that yes, it did. So for 17 patients, if we perform that same process in the British cohort, we again find that we do significantly better with our predictions if we computationally grow our lesions to the correct time point. And so in summary, I've shown that it's possible to effectively deploy language models, electronic academic scans from MRI scans to assess in English to acute clinical CT scans of stroke patients who are assessed in Spanish. And for this project, it was critical to establish that lesion growth must be appropriately accounted for when there is a large discordance between imaging and behavioral assessments. And another thing that I want to highlight is for this project, I developed an in-house MATLAB algorithm toolbox for segmenting stroke lesions in CT scans, and the lab still uses this toolbox to this day, and has now adopted it as their main framework for doing lesion identification for MRI scans addition. And so finally, I want to talk to you about kind of the real thing which I'm focusing on right now, which is looking at the genetic influence related to neurodegeneration and specifically to Parkinson's disease. So Parkinson's disease and iron dysregulation have been implicated in one another. Parkinson's disease is a neurodegenerative disease primarily affecting movement, affecting more than 10 million people globally. And the PD patients, when you look at their brains, both in vivo and post-mortem, seem to show evidence of iron deposition in motor circuits of the brain. So there seems to be, at the high iron end, there's some relation of iron and its risk for Parkinson's disease. But similarly, at the low iron end, there also seems to be risk for Parkinson's disease. So anemic patients and male blood donors seem to be more likely to suffer from Parkinson's disease. Additionally, a number of people have looked at dietary iron and they found that non-heme iron is linked with Parkinson's disease risk. And others have suggested that policies of iron fortification in cereal and baby formula last century may have led to an increase in prevalence of Parkinson's disease cases. And so in trying to understand and study this question, one might look at the brains of Parkinson's disease patients or blood donation policies or rates of anemia, as well as dietary iron. But we took an alternative approach, which is to look at a genetic variant that essentially leads to opening the floodgates for iron absorption in the body. And that leads to a disorder known as hemochromatosis, and it is a recessive genetic disorder which leads to excessive iron absorption overload. It's predominantly caused by 2 copies of a mutation known as C282Y. And although iron is essential for the body to function properly, excess iron can lead to oxidative stress and to damage to healthy tissue. For this genetic variant, it's been seen that there's a higher disease burden in males versus females. And it's thought that the reason for this is that females have, through menstruation and childbirth, a natural mechanism of getting rid of this excess iron, which men don't. And if left untreated, excess iron can cause damage to the liver and heart, causing liver cancer, increasing chance for liver cancer, heart complications, diabetes, and other life-threatening conditions. And a little interesting aside, the treatment for this disorder is somewhat medieval. So it's bloodletting. And so, yeah, it's basically phlebotomy. So you just remove some amount of blood, and that seems to be highly effective at reducing iron levels and reducing secondary disorders that result from this genetic mutation. And so a few studies before we conducted ours had looked at the neurological impact of hemochromatosis. These are still largely disputed. So some studies have shown that 2 copies of this mutation had imparted a higher risk for Alzheimer's or Parkinson's. Other studies reported no risk or even a protective effect. We thought these studies' conflicting results might be explained by the fact they have modest sample sizes. They've pooled analyses across sexes, which I think is maybe the most critically important thing. We know that this is a genetic variant that has a sex-dependent effect, and none of these previous studies accounted for the fact that there may be a differential effect in males versus females. And also they had biased subject attitude. So they recruited individuals and whether they had Alzheimer's disease or hemochromatosis. They didn't necessarily capture the subclinical cases of people who just harbored the mutation, may not have had a diagnosis to either. And so we sought to kind of ask this question in a much larger sample than previous studies, looking at the UK Biobank, which is a population of 500,000 individuals who are 40 or older, who've all been genotyped and with whom we have electronic health records. And approximately 40,000 of these have neuroimaging brain scans by the time we've done this analysis. In the whole sample, we had just short of 3,000 individuals who had 2 copies of this mutation. with 165 who had neuroimaging. So what we did was we found a ratio of 4:1 controls matched for age, sex, scanner, mean cortical area, and top 10 principal components, getting 671 healthy controls. And so what we really wanted to ask was, given these 165 who have this mutation, these 671, if we compare the brains of these individuals, can we find evidence of iron accumulating in certain areas? And so MRI sequences can be set up in multiple different ways to be sensitive to different tissue properties, but we specifically chose T2 weighted imaging, which is sensitive to iron accumulation, where lower T2 intensity indicates more iron deposition. And we essentially tried to predict T2 voxel-wise intensities from this mutation.
UK Biobank didn't have any susceptibility weighted imaging? Or
They have some available now, and so that's kind of one of the things that I want to look at in the future. But while we were doing this project, it was sort of just becoming available.
Thanks.
Yeah, and so if we compare the brains of these sets of individuals, those that have this mutation for excessive iron absorption and those that don't, we can look here to see this association map, which shows an axial slice. And it's what's being plotted as the Cohen's D, where blue regions indicate lower T2 weighted intensities, indicative of more iron deposition. And we see that the Cohen's D goes up to -1, indicating a pretty substantial effect, which is localized to the basal ganglia, including the putamen, thalamus, red nucleus, subthalamic nucleus, and dentate nucleus of the cerebellum. And so, given this pretty substantial and localized effect to critical motor regions, we wanted to ask the question, was this evidence of iron accumulation in these critical motor regions related to risk of developing movement disorders? We found that it was. So here on the x-axis is the odds ratio. So what is increased risk for individuals who have this mutation for developing any of the disorders on the y-axis, separated by males in blue and females in yellow. What we see is that males specifically have a nearly twofold increased risk for developing essential tremor and Parkinson's disease. So these two results, kind of this association map as well as this disease enrichment, we published in JAMA Neurology, in which I was first author a couple of years ago. And this kind of formed the basis for the next research question, which we wanted to ask, which was, given this really extreme case of iron disruption, could that inform us about something more generally about neurotypical individuals who maybe don't harbor this mutation? And so kind of the first question was, does this pattern of archetypal iron accumulation predict risk for movement disorders generally for individuals who don't have this mutation? And additionally, are there any other genetic or dietary routes leading to this pattern of iron accumulation? So in order to do that, we once again turned to UK Biobank, where we trained a classifier to differentiate between individuals who didn't have this mutation and did have this mutation from brain scans alone. And so this essentially creates classifier weight, which is in essence what I'd shown you on the previous slide, allowing us to differentiate between these two groups. And if we perform cross-validation within this cohort and evaluate how well we can differentiate between these individuals, we do fairly well here with an area under the curve of 0.86. So this is called a ROC analysis. And for those who aren't kind of maybe familiar with this sort of approach, you can it's kind of somewhat analogous to an accuracy. So for an accuracy of 100% says you're perfect at distinguishing these two groups. Here we have an AUC of 0.86, which you can somewhat sort of map on to an accuracy of 86%. And so kind of the next stage, what we decided to do is, given that we could do fairly well at differentiating individuals, given this mutation or not from brain imaging alone, kind of most interesting bit is we took these scans, these weights, and applied them to new scans to see, could we predict, could we use that to kind of generate a score that says how much these individuals exhibit the sort of archetypal iron accumulation that we observe for human chromatosis individuals. So from this, we generate a single score, which we call a polyvoxel score, which in essence captures this continuum of iron dysregulation in central regions of the brain. And then we ask the question, because sort of from this, we have a distribution which we refer to as the hemochromatosis brain liability, essentially capturing this degree of iron accumulation. And we wanted to ask in this independent sample, does that predict risk for neurological disorders? We find that yes, it does. So here on the x-axis is the odds ratio for that polyvoxel score predicting the disorders on the y-axis. We see that it predicts risk for Parkinson's disease in this independent sample of individuals who don't have this mutation. We also find that it predicts risk for abnormalities of gait and mobility. And so one thing which you might notice here is that it's a protective effect. So it's kind of going the opposite direction, which was a little bit unexpected at first, but we wanted to kind of understand this more closely. And so what we did was we did a couple of things, which was firstly, we separated the polyvoxel score into different quartiles. So the lowest 25%, the next 25%, the next 25%, the next 25%. And additionally, we used models that had been fitted on resected liver tissue, which maps MRI signal to ground truth measures of iron from this resected liver tissue. To kind of impute what we thought the estimated iron concentration would be for these critical regions that we use to create the score. So if we do that, we have on the x-axis the estimated brain iron concentration for these regions, and then each of these blue points indicate different quartiles. So the lowest 25%, the next 25%, the next 25%, and the highest 25%. And then on the y-axis, we have the disease enrichment for Parkinson's disease with odds ratio. And additionally, what we can do is we can introduce individuals who we use to train the classifier, those C2A2Y homozygotes, to see that they are both at the high iron end and also increased risk for Parkinson's disease. So we kind of seem to uncover this U-shaped relationship where there seems to be risk at both the low and high iron ends for disease enrichment for Parkinson's disease. And there's a reasonable amount of literature to support kind of both ends of the spectrum. So at the low iron end, as I mentioned earlier, there's anemic and male blood donors that have higher rates of Parkinson's disease. Lower hemoglobin levels is associated with greater Parkinson's disease severity. Low serum iron is observed in PD patients. And finally, therapy promoting the formation of red blood cells is shown to have neuroprotective effects in preclinical models. At the high iron end, it's been observed in post-mortem and in vivo imaging that there's more iron deposition in motor circuits of the brain. Neurodegeneration is a rare form of genetic disorder in which individuals with rare genetic mutations have excessive iron accumulation in motor regions of the brain, similar to the pattern I've shown you. And the most marked symptom profile for these individuals is profound Parkinsonian symptoms of early ages. And iron chelation, which is where you give individuals drugs that reduce iron levels, has shown some promise reducing the kind of symptom progression for these NBIA patients. And so moving on from this result, we wanted to see, given that it seems to predict risk for Parkinson's disease, we wanted to see, could this variability in iron accumulation, are there other genetic routes that might lead to this kind of iron accumulation? So I've shown you that this C2A2Y mutation leads to this localized pattern of iron accumulation that we use to create this score that kind of captures this continuum of iron accumulation in these critical motor regions. So next, we wanted to kind of perform a genome-wide association study to see, are there other genetic variants that could predict this variability in iron accumulation. And if we do that, we can look here at creating a Manhattan plot, which on the x-axis shows different locations in the genome, and the y-axis shows the negative log 10 p-value, which indicates how strongly each region is associated, each region of the genome is associated with this pattern of iron accumulation. And the red dotted line indicates bond-primary significance. So we find 43 genome-wide significant loci, and if we calculate something known as a SNP heritability, how much can we attribute this pattern of iron accumulation to genetics, we see that it's around 0.4, so about 40% determined by genetics. This quantity is also sample size dependent, and so for this sample size, that SNP heritability is considered very large. So it does seem to be very strongly determined by genetics. If we look at some of these peaks as well to see what genes they seem to be related to, We see that this top peak is in transferrin, which is the principal glycoprotein responsible for transporting iron around the body. Then we find this mutation HFE. So this is the same mutation that was used to train this classifier, but here it's the heterozygote individuals, so individuals that just have one copy of that mutation seem to also be related to this pattern of iron accumulation. Similarly, this mutation here is in the biological pathway that's responsible for producing hepcidin, which is the key hormonal regulator of iron absorption in the body. And then we also see these genes, which are known to be key regulators of iron transport, both in the body generally and also in the brain. And we wanted to take these results and use them in conjunction with a technique known as Mendelian randomization, which is a method that has emerged as a method for inferring causality from exposure to an outcome using observational genetic studies. And so to just kind of step aside from this project just for a moment, just to give some context of this method more generally, if we're interested in, say, the relationship between cholesterol and cardiovascular disease, which in observational studies has been established, there is a reasonable link there, but maybe we wanted to test to see, is there a causal relationship between these two? So in this context, we have our exposure, which is cholesterol, and our outcome, which is cardiovascular disease risk. And so if you wanted to test whether this relationship is causal, the gold standard measure for doing that would be to conduct a randomized control trial in which individuals given a placebo that doesn't change cholesterol or a drug that reduces cholesterol. And by manipulating this exposure, if we expect that there is a causal relationship between these two, then we would expect that the drug should have no effect on cardiovascular disease risk, but that it should reduce cardiovascular disease risk when we give a drug that reduces cholesterol. I think I might have got my words mixed up. But you get the point. Placebo is not going to do anything. The drug's going to reduce cholesterol and cardiovascular disease risk. And so this approach relies on randomly assigning individuals to an exposure group in order for us to have confidence there is a causal relationship between these two. And so to contrast that with Mendelian randomization, what we look at is genetic variants, some of which decrease cholesterol levels, some of which increase cholesterol levels. And if there is a causal relationship, what we would expect is that those have a downstream consequence of affecting the outcome where those that decrease cholesterol levels decrease cardiovascular disease, and those that increase cholesterol levels increase cardiovascular disease. And critically, this approach leverages the random assignments of genetic variants during meiosis. So bringing it back to the project that I'm describing, we wanted to test the relationship between peripheral blood markers to predict brain iron accumulation levels as captured by this polyvoxel score, which I've been showing you. And if we kind of perform this analysis, we see that there is fairly strong evidence for a causal relationship of peripheral blood markers to central iron markers in the brain. And so if we test the reverse, so if we condition on central iron levels, those genetic markers, and see does that causally predict peripheral blood markers, which we wouldn't expect it would, we see that there's a null effect, which is reassuring evidence that essentially there is a strong evidence for a causal relationship from peripheral blood iron markers to brain iron accumulation patterns, but there is poor evidence for the reverse, that brain iron levels increase peripheral iron. And so these are the results for serum iron. Similarly, if we perform the same analysis for ferritin, we see that there is a strong relationship and transferrin saturation as well as total iron binding capacity, where we see that serum iron and transferrin saturation show evidence for a strong causal relationship impacting this pattern of brain iron accumulation. So for the final project, I'm going to talk to you. Thank you for staying with me, by the way. For the final project, we wanted to look at are there any dietary patterns that could predict this pattern of brain iron accumulation. And so this was work which I supervised, which was led by a research assistant who we've been working with for a few years, his name is Jonathan Loughnan, and he's recently been accepted into the cognitive science department here at UCSD in his PhD program. So if you have any opportunity to work with him, I can't recommend him more highly. And so what he looked at was a number of food preferences in UK Biobank in which individuals score their preference toward or against certain foods. So it's 150 questions in which they're assessed, and 335, this includes 35,000 participants. And so you can imagine across these 150 questions, if people have a preference, say, towards cake, maybe they have a preference towards other sweets in general. And so what he first looked at was just the factor structure to see kind of how much overlap is there in terms of correlations for certain food preference questions. And here's a screen plot indicating that there is a reasonable amount of degeneracy in that. And so this is a busy plot, but I'm going to walk you through it. This is the correlation matrix across all of those 150 food preference measures. And it's kind of interesting when you look at it, it's kind of clustered and grouped according to their correlations. So we see kind of these two big blocks here and here, which largely map onto the kinds of things that we think of as more healthy sort of food preferences and less healthy food preferences. So if we look at the sub blocks within these, this first block relates to a preference towards seafood. This block relates to a preference towards exercising. This next block relates to a preference towards fruits, vegetables, and olive oil. And then if we look at this other block, we see that there's a preference towards fried food, towards red meat, a preference towards sweet foods in general, and a preference towards butter, chips, pasta, and pizza. And so from this, he created factors that were related to kind of each of these blocks of correlation that we saw. And then he associated those with this polyvoxel score of brain iron accumulation. And so when you perform this association, here on the x-axis is the effect size correlating that each of preferences towards each of those factors, on the y-axis with variability in this brain iron accumulation score. And so here I'm somewhat simply kind of annotating it in the direction of more and less iron. But you can see that savory food, red meat, and salty foods seem to be related to more brain iron accumulation, and that preferences towards sweets and fizzy drinks seem to be associated with less brain iron. And so we kind of wanted to bring this back to the clinical research question, which we're interested in, which is this pattern of brain iron accumulation related to Parkinson's disease risk, and can we also combine that with each of these food preferences? And so what he looked at here was looking at the joint effect of each of these food preferences with either the brain, so each data point is each of those food preferences, and then on the x-axis is association with the polyvoxel score, this brain iron accumulation metric that we have, and then on the y-axis is each food preferences association with Parkinson's disease risk. And so one thing which you'll note is that there's a significant negative relationship, which is what we were expecting. So I mentioned a few slides ago that there's this kind of U-shaped relationship with brain iron and Parkinson's disease risk. But what we have is at this high iron end, these individuals only represent about 1% of the population, whereas each of these blue points represents 25% of the population. So in this regime and what he's studying, we're largely looking at this kind of descending shape relationship. And so in that space, in kind of neurotypical individuals, what we expect is kind of a negative relationship between brain iron and Parkinson's disease risk. And if we highlight some of these quadrants, so here we see in the top left that preferences towards certain sweet foods, including cake icing, cookies, jam, and ice cream, seem to be related to lower brain iron levels and greater Parkinson's disease risk. And a preference towards alcohol seems to be associated with greater brain iron accumulation and lower Parkinson's disease levels. And so we wanted to kind of validate this to see, like, does this pattern that we saw in this meta-analytic approach, is that revalidated when we just looked at voxel-wise intensities? So he associated each of those food preference factors with voxel-wise T2-weighted intensities, where we again saw what was kind of shown in that previous scatter plot, where a preference towards sweets was associated with less iron, and a preferences towards alcohol seems to be associated with T2-weighted intensities consistent with more iron. And there is reasonable evidence to suggest at least peripheral markers are in this direction. So there are experiments in which individuals are given glucose pills or placebos, and they find that those they're given glucose have a causally decreased levels of iron. And they've shown that this is regulated through hepcidin production from the pancreas. And similarly, observational studies of chronic alcohol users have been shown to have higher levels of iron through the disruption of, again, hepcidin that's secreted from the liver, which is all consistent with what we are seeing. So in summary, I've shown you a project looking at this mutation, which leads to a localized pattern of iron accumulation and a male-specific increased risk of Parkinson's disease. I showed how this pattern of iron accumulation can be used to generate a score which captures a continuum of iron accumulation that uncovers a U-shaped relationship for Parkinson's disease risk. It's strongly determined by genetics, via peripheral blood markers that seem to show strong evidence of a causal relationship. And finally, that a preference towards certain dietary patterns seems to predict this variability as well. And so we think that the hemochromatosis spring may represent a Parkinson's disease imaging biomarker that's important for understanding a continuum of brain iron accumulation, potentially useful for patient stratification. And so, as I mentioned before, we published this in JAMA Neurology a couple of years ago. And then looking at the genetic architecture of this phenotype, we have a preprint available, which has just been accepted in the last couple of days to Science and Advances. And then I'm senior author on this paper looking at dietary patterns and how that relates to brain iron accumulation and risk of Parkinson's disease. We also produced code which allows individuals to, with 2 lines of code, check the registration of these weights to their brain imaging scans, and also create this polyvoxel score within their own data set. And so for future work, I'm hoping to evaluate this polyvoxel score biomarker in more PD-focused data sets to understand if they relate to any distinct clinical features. And then I think this work looking at showing that peripheral blood markers seem to be strongly causally related to brain iron accumulation patterns kind of suggests that we might be able to find a mapping from blood markers to central iron markers. And if such a mapping could exist, then we could potentially gain access to the central iron markers from just doing a cheap and easily administered blood test. And so there's certain data sets I want to work with to address that question. And then finally, there's evidence with rodent models that brain iron accumulation and alpha-synuclein seem to interact with one another. So alpha-synuclein is the critical proteinopathy implicated in Parkinson's disease risk. And there's evidence that that iron seems to interact with it. So I really want to evaluate how the interaction of these effects seem to affect disease progression for human models where we have access to both of these types of data. So this would be work that I will work under the mentorship of Irene Litwin. That kind of some of the ideas that I'm writing this KOL around. But I also will be interested in prospective collaborations within the department. And so I personally would like to work still within neurodegeneration. Personally, my mum was diagnosed with Alzheimer's a few years ago, and so that's something that's particularly close to home for me. And so I'd be very interested in working with Judy Parr, looking at some of the genotype environment interactions that she's been looking at, potentially extending some of this APOE work to look to see maybe if we could relate to the dietary food preferences that I've kind of shown previously. Also, some of the important work with David Coughlin, as well as Douglas Glasgow, looking at seeding amplification assays for here alpha-synuclein, but also amyloid beta, potentially see could we use these multivariate imaging prediction methods to, instead of predicting genotype, could we predict status on these seeding amplification assays to improve prediction of these patient groups. I think also I'd be interested in applying these to other imaging modalities, such as PET scans, maybe extending work with potentially Sarah Banks, looking at sex differences that we see in cognitive aging. And then additionally, I'm particularly interested in working with Hector Gonzalez in underserved communities such as Hispanic and Latino communities, where I'd be interested in working with the Salinka data set. And one of the interests there is that they measure these direct peripheral blood markers, which would be critical for kind of finding this mapping between peripheral blood markers and central blood markers in the brain. And I think more importantly, kind of making sure that we generalize these findings to other non-white populations. I think it's an important pressing need to make sure that we close health inequities that we see in medical research. So I've talked to you about a few projects looking at the genetic influence in adolescents related to behavior and disorders of the brain. I've then shown you a project which looks at how we can use brain imaging to predict language outcomes after individuals have experienced a stroke. And then finally, I've kind of talked to you about the sort of main research agenda, which I'm focusing on now, which is how we can use a genetic mutation which affects iron absorption to create an imaging biomarker that's predictive of risk for Parkinson's disease. So with that, I'd like to thank all of the study participants, the institutions that support this, as well as the funding agencies, and the very generous mentorship team that have supported all of this work. I'd like to, in addition, thank you all for listening and inviting me to questions.